Amyotrophic lateral sclerosis, you know it as ALS, is one of the biggest medical mysteries out there. But what if the disease we've been calling ALS isn't just one disease? What if it's actually two diseases in disguise? We're going to dive into this incredible neurological puzzle where two conditions look exactly the same to a doctor, but at a cellular level, they're fighting completely different battles.
So yeah, think about that for a second. What if everything we thought we knew about how to fight this thing was only half the story? This is the exact question at the heart of some groundbreaking new research. And the answer, well, it could change everything about how we understand and, more importantly, how we treat ALS.
All right, so let's meet our two main players. On one side, you've got sporadic ALS, or SALS. This is the one you hear about most often, right? It just seems to pop up out of nowhere, with no clear family history or genetic reason. And on the other side, we have C9 ALS. This one's different. We know exactly what causes it, a specific inherited mutation in a gene called C9orf72.
Now, here is the absolute core of the puzzle. Even though one is seemingly random and the other is purely genetic, for the person experiencing it and for the doctor diagnosing it, they look identical. The symptoms, how the disease progresses, you just can't tell them apart by looking at a patient. It's a medical mystery.
So to even begin to solve this, scientists had to go deeper. Way, way deeper. They had to look past the symptoms, zoom right into the brain itself, and focus on a very special group of cells. You could call them the brain's first responders.
Let me introduce you to the microglia. These guys are amazing. Think of them as the brain's own personal cleanup crew, security guards, and paramedics all rolled into one. In a healthy brain, they're always on patrol, looking for any sign of trouble, any damage, or any debris that needs clearing out. They are absolutely critical to keeping your nervous system running smoothly.
And these microglia basically have two modes. Their normal, everyday mode is what's called homeostatic. They're just calmly patrolling, keeping an eye on things. But the second they detect a problem, like the damage from ALS, they're supposed to flip a switch and go into reactive mode. The alarm bells go off, and they jump into action to fight the threat and clean up the mess. That's how it's supposed to work.
But this is where our two identical diseases suddenly start to look completely different. The huge discovery here is that in sporadic ALS versus C9 ALS, the brain's cleanup crew behaves in totally opposite ways.
And here it is, the key difference. In sporadic ALS, the microglia get the signal loud and clear. They power up, they go into that reactive state, and they launch a full-scale immune response, exactly like they're designed to do. But in C9 ALS, it's a different story. The response is just weak. The microglia hear the alarm, but for some reason, they just don't fully activate. They fail to make that all-important switch.
I mean, just look at the difference. In sporadic ALS, that activation level is high. The immune response is strong. In the genetic C9 ALS, it's seriously low. It's kind of like one team of firefighters hears the alarm and rushes to the scene with sirens blaring, while the other team just sort of stays in the firehouse, polishing the truck.
So, the big question is why? Why does the cleanup crew in C9 ALS basically call in sick? Well, the answer takes us right back to that one specific gene, C9orf72. You see, the mutation in this gene isn't just a small typo. It's more like a genetic stutter, a stuck record. A tiny bit of genetic code, GGGGC, gets repeated over and over and over again. We're talking hundreds, sometimes even thousands of times. This enormous, messy repetition is the defining feature of C9 ALS. And because of this stutter, the gene just can't work right. It's something called a loss of function. All those repeats basically jam up the works, so the gene can't produce enough of the protein it's supposed to make. It's not totally broken, but it's like an engine that's only running at half power. And it turns out the microglia really need that engine running at full blast.
So what does this loss of function actually do? Well, it directly cripples the cell's own internal garbage disposal system. This system has a fancy name, the endolysosomal pathway, but just think of it as the network of garbage trucks and recycling centers inside each and every microglial cell. When that pathway breaks down, the cleanup crew literally can't clean up. Their tools are broken.
And it gets worse. This isn't just a problem inside the microglia. This one failure creates a devastating ripple effect, taking down the entire support network that's supposed to protect the brain's motor neurons.
So, because the microglia aren't activating properly, other super important support cells, called astrocytes, also fail to get the message. They stay in their passive state too. The communication between all these critical support cells just falls apart, and what you're left with is a total system-wide failure, leaving those vital motor neurons completely exposed and vulnerable.
Okay, so we've gone from a clinical puzzle all the way down to a genetic stutter and a breakdown in the cell's garbage disposal. Let's zoom back out. Why does cracking this case matter so much in the real world?
This quote from the researchers who made the discovery really says it all. Even though the diseases look the same on the outside, what's happening on the inside, these cellular and molecular substrates, is fundamentally different. And knowing that, well, that changes the entire game.
This discovery unlocks a whole new, much smarter way to fight ALS. For starters, we can actually group patients based on what's truly happening inside their cells, not just by their symptoms. And that means we can develop targeted treatments. For sporadic ALS, maybe you need a drug to calm down that overactive immune response. But for C9 ALS, you need a drug that does the complete opposite, one that boosts the failing response. This lets us design better clinical trials, where we give the right drug to the right patients, which hugely increases the odds of finding treatments that actually work.
So we're left with this really powerful idea. Now that we know the cleanup crew in genetic ALS is broken, not overactive, the question becomes, could we find a way to reboot it, to get those microglia back online and doing their jobs? It's a total shift in strategy, from trying to fight the inflammation to trying to fix the failure. And that, right there, is a brand new, very hopeful path forward in the fight against ALS.